Good afternoon, everyone, and thank you for joining us for today's webinar. This webinar is the third webinar in our FTCOE webinar series, The Opioid Crisis, a Public Health Enemy. Rates of opioid use and misuse have reached epidemic proportions and are impacting many aspects of both criminal justice and forensic science programs. Opioid addiction is the driving force behind this increase, in use with nearly 3 million Americans reporting a substance use disorder to prescription pain relievers or heroin in 2015, fueling a steady increase in fatalities to an estimated 91 U.S. deaths daily. These rates are not slowing, rather they are doubling, quadrupling, or undergoing even more alarming increases in some areas. Law enforcement, medical professionals, laboratories, and legal agencies are battling with unmanageable caseloads, economic shortfalls, and challenges for safety, analytical preparedness, and basic education and training. Reliable surveillance and intelligence are needed more than they have ever been, confronted with the fast-paced emerging drug life cycles. This legislative quagmire is just as burdensome, as policy change cannot happen without the data to support that change. This webinar series will bring a multifaceted perspective to how diverse criminal justice disciplines are addressing these challenges, sharing their knowledge, and advancing science, technology, and law. Dealing with the impacts of the opioid crisis to the criminal justice system requires better reporting, surveillance, research, technology, and policy than are currently in use. The need to understand the epidemic and its effects goes beyond knowing your own profession. It takes a global perspective to fully act and make a difference. My name is Josh Vickers, and I will be your technical host today from RTI and the Forensic Technology Center of Excellence. Today's webinar is titled Fentalogs, Pharmacology, Toxicology, and Analytical Approaches. Your presenter for today is the esteemed Dr. Barry Logan. We are very thankful to have him on today. Dr. Logan is the Senior Vice President of Forensic Science Initiatives, Chief Scientist at NMS Labs in Willow Grove, Pennsylvania, where his responsibilities include management of toxicology resources, new test design and development, and expert testimony in forensic toxicology and chemistry. Dr. Logan is a fellow of the American Board of Forensic Toxicologists and has over 100 publications and 400 presentations in forensic toxicology and analytical chemistry, including work on the effects of methamphetamine, cocaine, and marijuana on drivers, drug-caused, and related death. In recognition of his work and contributions, Dr. Logan has received numerous national and international awards, and in 2013 served as the president of the American Academy of Forensic Sciences, or AAFS. Just our quick disclaimer, the opinions, findings, and conclusions or recommendations expressed in this presentation are those of the presenter and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. And now that we have got our disclaimer out of the way, I would like to go ahead and switch things over to our presentation today and hand things over to Dr. Barry Logan.
Thank you, Josh. Welcome, everybody. It's a pleasure to see so many of my friends and colleagues logged on. I see a number of our colleagues from Brazil are joining the webinar today, too, so welcome to all of you. The focus of my presentation is going to be about the fentanyl class. This is a class of drugs that's taking up a lot of our time here at NMS Labs currently. Because it's starting to see the same kind of turnover in terms of numbers of compounds that we historically saw with drugs like the synthetic cannabinoids. So just preliminarily, I will be talking about some of the testing that's performed at NMS Labs by way of disclosure, but I'll also be talking about some testing that we perform at our nonprofit Research and Education Foundation Center for Forensic Science Research and Education. And I'll also be mentioning one particular test performed at the center on a piece of equipment that was loaned to us by Waters Corporation. So the objectives today are to talk about the class of the fentanyls, both those that are prescription and illicit, they are coming out with such rapidity that it does get kind of confusing to keep track of them. So hopefully we can clarify just what the different structural classes are. Then we'll talk about their toxicology a little bit, and then consider the approaches that we're using in our respective laboratories for screening methodologies and for confirmation of fentanyl analogs. And I'll talk just at the end about some of the challenges in terms of keeping up with current scope and trying to make sense of what those results mean once we go to the trouble of detecting and measuring these compounds. So I borrowed this term Fentalogs from the internet. I didn't invent it, but it is something that's popular on some of the drug user forums, and it's what's called a portmanteau word, where you take different pieces of other words and make a new word, so like chillax and so on. But here we're talking about Fentalogs. Fentalogs a little easier on the tongue, only 3 syllables instead of 6. But it just reinforces the idea that we're talking about things that are derived or related to fentanyl, that the various members of the class have structural features that they share with that parent compound. Fentanyl itself was invented in the 1960s by Dr. Paul Janssen, who was a prolific drug designer, created his own pharmaceutical company in Belgium, and over the course of his career made many thousands of different therapeutic drugs, both in the narcotic analgesic field, but also anticonvulsants, antipsychotics, antifungal drugs. But he's probably best known, and the Janssen Pharmaceutical Company is probably best known for fentanyl and some of the related narcotics that it developed. The structure-activity relationship really started with the consideration of meperidine, which is up in the top of the slide here, and looking at the proximity of various functional groups and how they're separated spatially led to one of Dr. Janssen's first compounds, which was dextromoramide. Dextromoramide became a very widely used narcotic analgesic drug around the world, one time used in as many as 50 countries, and had a potency of greater than that of morphine. With some further experimentation, he developed another drug called pyritramide, and you can see kind of the evolution towards the fentanyl structure here. We have the piperidine ring. Here we have the amide functional group up here, and then another piperidine ring here, a carbon chain that separates the piperidine ring from a phenyl group or aromatic group. And then that led ultimately to, amongst many other compounds, the development of fentanyl itself. And we'll talk in a little more detail about the structural elements of fentanyl. Originally, in the patents that described it, it was called R4263, and sometimes you'll see fentanyl referred to as that. So here is the fentanyl molecule. We'll look at the three basic structural components. There's this phenethylpiperidine moiety here, and in almost all of the compounds we'll talk about, this is a 2-carbon chain that separates the piperidine ring from the phenyl ring with the alpha and beta positions. So when we talk about things like alpha-methylfentanyl, beta-hydroxythiofentanyl, that's where those substitutions take place, and we'll look at those examples. But this part of the molecule can be modified by either changing the length of this chain, by substituting in the alpha or beta positions, by substituting on the phenyl group, or by substitution on the piperidine ring itself. So you're already starting to get the sense of all of the various different combinations or structural modifications that could make this a very large class of drugs. So for example, with this moiety, if we substitute the phenyl ring at the end here with a thionyl group, we change the compound. In the case of phenylfentanyl, we've done two things. We've actually shortened this to a single carbon unit, and shortening that carbon chain actually makes the compound, to all intents and purposes, inactive. So we rarely see fentanyl analogs that have just a single carbon atom in this linkage. And phenylfentanyl and related benzylfentanyl, where we've retained the phenyl group here, were on the federal schedule of Schedule 1 drugs for a number of years, but in 2010 were removed from the schedule because studies had demonstrated their lack of activity. So it seems to be that having the 2 carbon units in this chain here is critical to their functionality. But certainly any other aromatic group that's placed at the end of the chain, as we'll see in some other examples, does allow the compound to retain its activity. Again, substitution in the of the phenyl group with the thionyl group is thiofentanyl, and then in the beta position here, equivalent beta position, we have hydroxy. So beta-hydroxythiofentanyl was one of the more popular fentanyls among drug users back in the first wave of fentanyl use back in the 1980s. Substitution on the piperidine ring itself. So this would be the 1, 2, 3 position. So we have 3 thiofentanyl because we have the thionyl group here. So that's one other example. And then moving on to the other components of the molecule, the propanamide functional group. So 3 carbon atoms, which makes it propanamide. This is the chemical name here for fentanyl. Propanamide is reflected there. But by changing the length of the chain on the amide, or by adding other substituents to that chain, we can make other whole families of fentanyl-related compounds. So for example, replacing this chain here, the orientation is a little different, but in place of these 2 carbon units here, we have this isobutyryl group, gives us isobutyryl fentanyl. Putting this cyclopropyl group on gives us one of the more recently emerging fentanyls. Caused a series of deaths and illnesses down in Georgia just about a month ago, and is starting to emerge in our casework, but cyclopropyl fentanyl is substituted here. Acrylfentanyl also substituted in the propanamide, in place of the propanamide. And here we've replaced the chain, or the an aliphatic group, with this aromatic group. We've added furanol group on here to create 2-furanylfentanyl. As we'll discuss, 2-furanylfentanyl is currently scheduled. There are, however, also other structural isomers of this. We'll also mention 3-furanylfentanyl, which links the furanyl group to the rest of the molecule through the 3 position rather than the 2 position. And then finally, this other aromatic moiety, the phenyl group in all of the fentanyl compounds that I'm familiar with and have looked at is always present. However, there can be substituents that are added to that, as in this example where we've added this methoxy group onto the 4 position. 1, 2, 3, 4. And we have the butyryl group here in place of the propyl group, which is characteristic of fentanyl. So this gives us 4-methoxy-butyrylfentanyl. And then here in the 1, 2, 3 position, we've added a fluorine onto the structure of fentanyl, which is 3-fluorofentanyl. There is a homologous series that are substituted over on this side that I forgot to mention. Fentanyl itself is has a propyl group with the 3 carbon atoms. If we take one of these off, that gives us acetylfentanyl. So people often think that acetylfentanyl is addition of acetyl group to fentanyl. In fact, it's not. It just reflects the removal of one of these carbons. The 3 carbon version is fentanyl itself. The 4 carbon version is butyrylfentanyl. And then the 5 carbon chain creates valorylfentanyl, which is also one of the analogs that we're starting to see in our casework. So that gives you some flavor of the fact that there are hundreds of possible variants of the molecule, and it seems to be pretty robust in terms of retaining and even increasing its potency depending on where the substituents are placed. The drugs are, by virtue of their potency and their potential for addiction, they're heavily scheduled. Under the 1961 United Nations Single Convention on Narcotic Drugs and some subsequent modifications to that, the World Health Organization runs an expert committee on drug dependence that advises the United Nations on international scheduling. And signatories to the 1961 international schedule are those countries are required to adopt the scheduling agreed upon by the United Nations into their own country's schedules. So all of the compounds on this list here, currently there are 15 controlled. The most recently being added was butyrylfentanyl, which was added just this summer. And at the same time butyrylfentanyl was scheduled, the United Nations also added the two precursors, 4-ANPP and NPP, as scheduled compounds. 4-ANPP we'll look at a little later. In addition to being a precursor chemical, it is a minor metabolite of some of the fentanyl compounds. In addition to the compounds that are scheduled, the United Nations Office on Drugs and Crime maintains a list of compounds that they're aware of as appearing in member countries. Butyrylfentanyl was on this list. It just got moved to the actual United Nations list in the summer. But there are 19 compounds that remain on this list that countries are advised to monitor for and collect data which will support broader scheduling of those emerging compounds. So in the United States, since we are a signatory to the 1961 convention, all of the compounds, the United Nations compounds, are currently scheduled in the US. And in addition to that, the United States, through the Drug Enforcement Agency, has scheduled an additional 4 compounds. So icofentanyl, 4-fluoroisobutyrylfentanyl, carfentanil, and 2-furanylfentanyl are not currently on the UN list, but have been placed on the schedule through the publication of notice in the Federal Register by the Drug Enforcement Administration. And this is our own list here at NMS Labs of compounds that we are monitoring, either that we're starting to see in some of our ceased drug casework, or that we're seeing chatter about online, or we've learned from our colleagues that these have shown up in their casework, either in the United States or around the world. And again, it just reinforces the fact that the list of compounds that we have to be looking for and have to be aware of is constantly changing. And you can see patterns in terms of the variants that the illicit drug manufacturers are making. They'll take one compound as a family, like furanylfentanyl, and then make further modifications to that, altering either the chain length on the amide portion of the molecule or adding substituents to the phenyl ring. So moving on a little to talk about the pharmacology of the drug. For those of us who've been used to dealing with other opioids for many years, its pharmacology is very similar, interacts at the same 3 opioid receptors. So taking fentanyl as the prototypical compound, its interaction with the mu receptor causes the analgesia for which the compound was originally developed, and then some of the subsidiary side effects we associate with other opioids, pupillary constriction, constipation, sedation, but also the euphoria that encourages abuse and dependence are associated with the mu receptor, as is the dose-related respiratory depression. And as we'll talk about in a moment, that really is the major toxic effect of all opioids, but the fentanyls in particular. The kappa effects also contribute to analgesia, cognitive impairment, and so on. They also are best known probably for their dissociative and hallucinogenic effects. So opioid-related drugs and members of the fentanyl class that are more kappa active tend to be less popular with users because of the negative dissociative side effect. And then the delta receptor system also has some contribution to sedation and motor effects. The degree to which the drugs bind and are retained by the receptor, the rate at which they bind and then come off, so K on and K off at the receptor, influences their potency and also the degree to which they're likely to be displaced by other compounds such as the naloxone, the opioid antidote. So it's very difficult to find a single source of information about relative analgesic potency. Some of this work comes from the very early work that was done by Janssen himself that has been reported by other workers. Some has subsequently been done independently. But basically every research group is using a slightly different model, and they're all pretty much using animal models, particularly for these fentanyl analogs that have never been approved for human use. So these, the analgesic potency information here is a good rule of thumb, but in terms of its quantitative accuracy, in terms of dose calculations and so on in human beings, we have to be careful about that, and that's definitely done more on the basis of clinical management than just by the numbers. So each group uses maybe a different type of animal. They may use a different pain stimulus. They may have a different way of scoring the test. So it's not necessarily possible to have a true quantitative comparison between all of these different compounds. In addition, it's important to remember that this is one measure. So it typically, again, in an animal model, it's a measure of analgesic effect. And the degree of analgesic potency relative to morphine doesn't necessarily translate into the degree of, for example, respiratory depression. So as a rule of thumb, the more potent the drug, the more toxic it's likely to be. The lower the dose is necessary to produce toxic effects. But just exercise some caution in terms of making these quantitative comparisons. There is information hidden about many of these compounds and their relative potency in the older patent literature, and also in some work that was done by NIH, or at least sponsored by NIH at the University of Virginia and University of Michigan. But as I said, it's not easily accessible. So the in terms of the toxicology of the drug, the principal negative effect is respiratory depression, which is treated by the administration of an antagonist like naloxone. There are other antagonists, nalorphine and naltrexone, but currently only naloxone is approved for reversal in terms of treating an acute opioid overdose in human beings. This is a protocol. It comes from a nice overview of management of opioid analgesic overdose by Ed Boyer. And it describes the protocol that's used to progressively increase the dose of naloxone in the treatment of patients who are unconscious in respiratory depression. There is a lot of discussion about whether some opioid analgesics, and some of the fentanyl analogs in particular, are less amenable to naloxone reversal. So there's still some debate amongst that, about that amongst clinicians. Certainly the potency of the drug means that if you're taking a lot of it, it would require more naloxone to displace it. Also, the half-life of some of these analogs is substantially longer than fentanyl itself or morphine. Carfentanil, again, there's limited data from animal models, but it suggests maybe as much as 7-hour half-life compared to closer to an hour for fentanyl itself. So there's the risk that when the naloxone is administered, since the naloxone itself has a half-life of 1 to 3 hours, that it can be metabolized and removed from the system while the concentration of the drug that precipitated the respiratory depression is still significantly elevated. So that can require re-administration of naloxone for a longer period of time. Alternatively, in some circumstances, a clinician may elect to intubate and support respiration that way. When you do administer naloxone to an individual who is in opioid toxicity, you can precipitate a rapid withdrawal, which is why we see that recommendation for progressive incremental increases in dose, because you want to try to manage the withdrawal symptoms, because it can be quite unpleasant for the subject. Naloxone itself, as long as you don't have any opioids in your system, is relatively inert. So it's very frequently given in an emergency room setting if there's even a small risk that the individual's condition can be accounted for by ingestion of an opioid agonist. So in non-opioid using subjects, it's quite safe. We see I get a lot of calls and questions about what are the risks both to laboratorians as well as to first responders, law enforcement, EMTs, in terms of accidental exposure to some of these very potent fentanyl analogs. This is one example of a story from May that most of you probably heard. It did get quite extensive media coverage. There's a story about a police officer who was involved in an arrest. Subsequent to the arrest, one of his colleagues pointed out that he had some apparent white powder on his uniform. He brushed it off with his hand, and then as he thought about it and thought about how it got there, started to feel some effects, started to feel drowsy, believed he was having trouble with breathing, and was taken to the emergency room. So that story went viral on the internet and created all kinds of alarm and concern amongst first responders, laboratory staff, laboratory management about what risks people working around these drugs might be exposed to. Once people started actually looking at what the actual risks are and bringing a little more balance to that, certainly these can be dangerous compounds if mishandled and in the wrong kind of environment. If there's accidental exposure, the consequences can be serious. But Dr. Boyer, whose article I mentioned earlier, was interviewed for this article in Slate that provided some, I think, better perspective on how to avoid any risk of toxic exposure. And just about a month ago, or in fact less than that, July 12th, the American College of Medical Toxicologists and the American Association of Clinical Toxicologists issued a position statement on preventing occupational fentanyl and fentanyl analog exposure to emergency responders. And the link is on this slide. And they concluded that based on a survey of emergency room doctors and calls to poison centers, that they had not seen viable reports of emergency responders truly developing signs or symptoms that were consistent with opioid toxicity from incidental contact with opioids. And they go on to discuss the fact that incidental dermal absorption is unlikely to cause opioid toxicity. They give some of the pharmacological and pharmacokinetic basis for that. And they do suggest that routine handling of for drug evidence using nitrile gloves is going to provide sufficient dermal protection. They go on to say that in exceptional circumstances where There may be dust clouds if a package has been dropped or a package has been opened and there's a dust cloud, or there are droplets of drug suspended in the air, that could present a risk for clinical exposure or exposure resulting in clinical effects. However, that a standard N95 respirator would provide sufficient protection to somebody with even that quite dramatic exposure. The rationale for some of this advice is that when you think about transdermal absorption of fentanyl from patches, it takes somewhere from 3 to 13 hours for that drug to penetrate the skin, the fat, get into the circulatory system, and then ultimately 35 hours to reach peak concentration. So casual surface contact that's removed relatively rapidly is unlikely to cause opioid toxicity. They do give some general advice and precautions for sensible management of exposure, but it's really standard use of standard personal protective equipment. They say workers who may encounter fentanyl or analogs should be trained to recognize symptoms and have naloxone ready. That's good prophylactic advice. However, they make the good point that I think needs to be reinforced, just providing some context for the true risks. For opioid toxicity to occur, you have to get the drug and sufficient quantities of it into the blood from the environment, and it can't happen just from being in the environment of the drug or being around somebody who's ingested the drug. So they say for routine handling, nitrile gloves, situations where an enclosed space is heavily contaminated with a highly potent opioid, certainly water-resistant coveralls should be worn. So a lot of white powder around a laboratory that's involved in packaging or stepping down drug for sale is a higher risk environment. And they point out that any drug exposure should be removed with copious amounts of water, not with alcohol-based sanitizers, since these can accelerate the rate of dermal absorption of a drug. So moving on to some of the analytical considerations. I gave a webinar through RTI's program earlier in the spring where we were talking about TOF screening, and we use a TOF-based approach for our drug screening, and we've had to adapt and update that as the universe of opioids has increased. So if you want some more details on our process, please go back and review that previous webinar, because I'm not going to spend too much time on it. But to stay current, to know what's actually out there, what you ought to have in your scope for testing, does require some investment of time, reviewing the literature, talking to your colleagues, looking at what drugs are being scheduled by the government, reading some of the poison control center reports. We also spend a lot of time monitoring what's being discussed in drug user forums. We look at a number of international venues to see some of the drugs that are becoming popular in other parts of the world. And then we look for commonalities in all of these different intelligence sources, and that helps guide us in terms of where we're going to invest our R&D dollars in terms of updating and improving our scopes. So what has all that led to? This is an approach we've applied from 2008 when we started developing tests for synthetic cannabinoids and cathinones through to 2016, 2017. So 2016, I'll show you some of our prevalence data, really started to see an increase both in the number and diversity of fentanyl analogs, and that has continued through 2017. So our screening procedure, again, I go into it in a lot more detail in the previous webinar. We're running time of flight. This is not a QTOF method, but straight TOF on an Agilent platform with about an 8-minute runtime, and sample preparation is single-step liquid-liquid extraction. So the data analysis is really the key to being able to find some of these emerging compounds. We have 4 different levels of data review that we do. We have a defined scope that I'll describe for you. that is the advertised scope of our test, so our clients know what we're able to pick up. On top of that, we do run an additional review for the presence of newly emerging compounds. And as time goes on, as things become more established, we will make the investment in validating them for addition to the defined scope, so they become part of the routine scope. But having the expanded scope allows us to rapidly add new compounds with a preliminary validation before it is fully validated as a routine method. But certainly we never add something to our scope before we have a confirmatory method, an independent confirmatory method for it. The third tier is to use the TOF data for scouting. So that's one of the intelligence sources that we monitor for the presence of newly emerging compounds. Once we have a standard for them, then we would move them into our out-of-scope findings or expanded scope data review. And then I previously also discussed the use of TOF data for retrospective data mining. That allows you actually to go back in time if you archive your TOF data. And if we have a cyclopentyl fentanyl case today, we can go back and data mine our data for the last couple of years to see when the first time that appeared might have been, even though we weren't looking for it at the time. So I'm going to talk more specifically about the expanded scope that we're currently using for looking for these emerging fentanyl compounds. In our defined scope, we have 254 compounds with the classic drug categories. And in the emerging drug scope, the secondary database that we use to query the TOF data, we have an additional 75 compounds that we're currently monitoring for. And again, the benefit of having the supplemental scope is it allows for much more rapid updating of the scope without revalidating the entire method. So currently in our Tier 1 scope, we're looking for acetylfentanyl, alfentanyl, fentanyl, norfentanyl, and sufentanyl. And then as of today, we are looking for these additional fentanyl analogs. And we do review this on a monthly basis. So cyclopropylfentanyl we added quite recently, and then cyclopentylfentanyl we've just added in the last few days. So what does that look like in terms of our reporting? So here's an example of a case that would be tested according to our routine scope, that 270 or so compounds that we're routinely looking for. In this particular case, we had an alprazolam positive result on the initial data analysis. But then when the supplemental database is run that contains the most recently identified compounds, we found these additional compounds. So initially only alprazolam was detected, but with the additional database, we picked up methoxyacetylfentanyl and despropionyl fentanyl. Despropionyl fentanyl is a common metabolite of several opioids, so anything that is substituted only on the section of the propanamide portion of the molecule is going to give you despropionyl fentanyl metabolite. So that's a good way also to identify the emergence of new compounds that are substituted just on this portion of the molecule. If you see that metabolite, gives you some idea of where they're substituted. So we're evaluating based on retention time difference, mass accuracy, and we have a minimum response threshold that's required to report compounds out of this supplemental scope. So for our confirmatory method, this is actually a method that's used at the foundation for some of our research projects. We do partner with many medical examiners and coroners' offices to do case studies, case reports that we share either in publications or through professional meetings. Sometimes we'll go back retrospectively and test a series of patients or subjects. We'll test multiple tissues that perhaps weren't part of the initial analysis, so it's more of a research focus than a case reporting focus. For our confirmatory process, once the TOS screen is complete, this is a method that one of our staff, Melissa Trisha, has been working on. So it's quite comprehensive. It matches the scope of the schedules I showed you earlier, and it takes into account the emerging compounds that are not currently scheduled, but that we're most concerned about in terms of their likelihood for entering into the market. So this method is run on Waters TQS micro, which gives us the appropriate levels of sensitivity. We're typically looking down at 0.05 to 0.1 nanograms per mil as a reporting limit for this test. And really, because of the potency of some of these compounds, carfentanil, low fentanyl, fentanyl, that can be many thousands of times the potency of morphine, you really do need that additional level of sensitivity. So GCMS really is not appropriate anymore for screening for or confirming the presence of some of these novel fentanyls just because of their potency. So this method, fairly straightforward. I believe it's also about an 8-minute runtime. I'll show you a chromatogram in a few minutes. For this particular confirmatory test, we do use a clean screen, UCT clean screen column for sample preparation. The assay, of course, is validated, and we put quite a lot of effort into our validation procedures. We'll do matrix matching within run and between run precision, recovery, a whole range or battery of stability experiments as recommended in the SWIG-TOX guidelines. We'll look at dilution integrity, because many times we'll find compounds that are above or outside of our linear range. And then comprehensive interference and carryover studies as part of the validation protocol. So this is an example of the chromatogram from Melissa's method. So the last compound here, cyclopentyl fentanyl, is out about 6.1 minutes. But all told, I believe there are 19 compounds in this conformation. So starting with norfentanyl, methoxyacetyl fentanyl. We have some co-eluting compounds, 4-ANPP, tetrahydrofuranol fentanyl, and beta-hydroxyfentanyl are all under this peak. But they're resolved since they have independent, they're resolved by mass since they have independent transitions. Cyclopropylfentanyl is I believe here. And then cyclopentyl fentanyl is the last eluting compound in the chromatogram, quite a ways out, which is responsible for the end of the run. Performance on this system is excellent. Routine R-squareds of greater than 0.99 out of whole blood. And this is just two examples for methoxyacetyl fentanyl and cyclopropylfentanyl. So what are we seeing as a result of both selecting the appropriate scope and then applying those, developing, validating, and applying those methods to the analysis of casework? So this is data from NMS Labs post-mortem findings from medical legal death investigation cases, and this is just since January of 2016. So looking first at the major opioids, we have fentanyl, and as you can see from this, positivity or prevalence of fentanyl in our post-mortem casework, medical legal death investigation casework, is substantially higher than that from the heroin marker 6-monoacetyl-morphine. So fentanyl in these opioid deaths is by far the most commonly encountered compound. And then acetylfentanyl, we'll talk a little bit more about. In terms of the fentanyl analogs or Fentalogs, it was one of the first to appear. Actually, a small batch appeared a number of years ago in Michigan. It was responsible for a number of deaths. It disappeared for a while and then re-emerged in 2015. but it's somewhere around probably 10% of the frequency of the fentanyl cases. So a significant prevalence and ought to be part of any drug screening scope. Looking at some of the more esoteric findings from the compounds that I listed earlier, this scale is a little different, of course, because we've taken fentanyl and 6-MAM off just so it's a little easier to see. So this is acetylfentanyl. You can see that we had a peak or a spike of that back in March of 2016. So in terms of how rapidly this market has diversified, you can see we got a little bit of butyrylfentanyl, which is this green line. At the same time, the acetylfentanyl peaked. Butyrylfentanyl pretty much disappeared for a while, and then acetylfentanyl started to decline in popularity. And then towards the fall of last year, we made some updates to our method, and you start to see the increase in an appearance of a number of these other compounds. So this is furanylfentanyl here, this light blue line. So it peaked in terms of prevalence back in the spring of this year, March or April, and has now started to decline a little bit. Acetylfentanyl, you can see here, is on the upswing again, so seems to be a greater amount of that in circulation. This is 4-NPP. As I said, it's a common metabolite to many of these fentanyls that are substituted only on the propanamide section of the molecule. So it can derive from a number of these other fentanyl analogs, but it's an important marker for fentanyl use and ought to be part of your scope as well. Carfentanil, again, started really to appear in the fall of last year. We had our first case actually back in July, and then that we found by data mining. And then once it was part of our scope in the latter part of the year, and for this particular test code, started to increase, peaked around January of this year, declined a little bit, but just in the last few months up through June, seems to be on the increase again. U47-700, another opioid analgesic that I've talked about before, not fentanyl related, different structural class, but just in terms of context and prevalence, that's where it falls. Dark blue line is acrylfentanyl, which reached a peak in March and it seems to be on the decline a little bit. And then butyrylfentanyl, as I mentioned, first appeared in a few cases back in the spring of last year, but started to appear with a little more frequency in March and April of this year. So it gives you an idea of how the market is changing and the relative prevalence of these targets. This is just a summary of the total numbers of positives between January 2016 and January 2017 in our death investigation casework. So almost 14,000 fentanyl cases, which is quite incredible. We hear a lot of stories about the fentanyl, the opioid epidemic, and this is really a bellwether of that. 6-MAM, heroin marker, around 7,000 cases. And then lower prevalence, but very diverse group of compounds that make up the remainder of these positives. If you look at all of these compounds, 52% of our positives are due to fentanyl, 26% are due to 6-MAM. So in total, that would be, 78%. So the other 22% of our opioid, emerging opioid positives are as a result of the combination of these other compounds. And if we take fentanyl and 6-MAM out of the mix and look at the relative prevalence of these compounds compared to 4-NPP, this just makes it a little easier to see their relative prevalence. So the challenges in terms of what laboratories are facing, of course, one is once you've performed this analysis, you've quantified the result, how do you know what to do with that result? What do you compare that number to? And there is some debate about the value of quantifying NPS compounds, partly because of the effort it takes to validate and maintain a quantitative method, issues about the availability of reference standards, which we'll talk about next. But it's kind of a chicken and egg situation, and I certainly advocate for people to be as comprehensive as they can, to quantify when possible, and then to share and report that information, because that forms the basis for being able to make comparisons and interpretations in future cases. We did publish a review with the support of RTI in January of this year in Journal of Analytical Toxicology on toxicologically confirmed adverse events associated with novel psychoactive substances between 2013 and 2016. And that does include a section on the novel opioids, where we collected and consolidated published information about concentrations of these drugs in forensic casework. So I'd refer you to that as a resource. The other challenge, of course, is analytical standards. We do not report anything without having an analytical standard for it, and then correspondingly labeled internal standards for these quantitative tests. So we certainly rely on commercial vendors. We're in touch with the manufacturers about what we're finding, what we think are emerging compounds. They're pretty good at being current, we've found. Sometimes it can take 3 to 6 months depending on the compound. And then sometimes, particularly for drug metabolites, which many laboratories are not testing for, it's just not worth the commercial vendors' time to manufacture and market those compounds. So sometimes these times are never available unless you're going to go to custom synthesis. That is expensive. We've paid as much as about $10,000 for the synthesis of a couple of milligrams of a custom standard, so it's not something we can do for every compound that comes along. It can be a little more rapid. Sometimes we can get it in 1 to 2 months, but certainly they're not available on a commercial scale. Importation of drug standards excuse me, from international suppliers sometimes can be up to 2 to 4 months, even if they have it available, just to get it imported. And then I know from some of my international colleagues, particularly my friends in Brazil, about how difficult it can be when standards are available to import them into other countries, sometimes taking as much as a year or more, which given the cycle time of these drugs just isn't a feasible way to keep up with the problem. So just to wrap everything up for today, covered a lot of ground, of course, but hopefully you have a good feeling for the ways in which the fentanyl molecule is modified in order to fine-tune the potency, to try to stay a step ahead of the scheduling events in different countries. Inevitably, scheduling does lag a little behind what is in the market, and the newly emerging drugs can sometimes take 3 to 6 months before they can be temporarily scheduled, and then up to a couple of years before there's enough information to have them permanently scheduled. In terms of risks to laboratorians or to first responders, I think the consensus amongst the clinical and medical toxicologists is that if you use some common sense practices and protections, that the risk is relatively low. But to avoid situations where somebody's exposed to a cloud of dust, for example. From the laboratory point of view, it's obviously critical, given, again, the rapid emergence of new compounds, to manage your scope, to keep current based on what you're reading about, what you're hearing from NIFLS or from other sources. Keep engaged with the toxicology community, and try to keep your methods up to scope, up to par with what is known to be on the street. And then finally, interpret the data with care in the absence of reference data, but it's always worthwhile, I think, having quantitative data that can be shared with and be a resource to the community. So with that, I'd just like to acknowledge my colleagues at NMS Labs, at the Center for Forensic Science Research and Education, my colleague MJ Menendez at the Organized Crime Drug Enforcement task force for some of the information she provided on emerging compounds. And with that, I think we have a couple of minutes to try and address some questions.
Oh, yes, sir, Dr. Logan. We have quite a few questions that came in. Before we get into that, I'm just going to make mention of the pods down there at the very bottom of your screen. We do have our presenter contact information there for Dr. Logan, so make sure to take that down. The middle pod down there is our file share pod. It does have a PDF version of today's slides. So you're more than welcome to go ahead and download that now directly from the room. Simply click on the title and then hit the download files button in the very bottom of that pod. And then also in the bottom right hand corner, you have the two links that were on Dr. Logan's final slide. Simply click on the name of the web link and then hit browse to, and it will open that page for you. So the very first question we have, Dr. Logan, came around slide 38. You had made mention of a previous presentation that you had done. Is there a way to get that presentation, maybe if they email you directly or something along those lines?
So that presentation was, we did that as part of, I believe it was Agilent series with RTI, so I believe it should be on your website.
Okay. Agilent is actually hosting that for us. So if you go to Agilent's website, then that presentation that Dr. Logan is referring to should be on Agilent's website. It was, I believe, just a couple of months ago. Is that correct?
It was, yes. Yeah.
Okay. So our first question that we have have you seen what Kamen has named allyl fentanyl, which is similar to acryl fentanyl, but has an extra carbon in the chain prior to the double bond?
I haven't looked at the structures on Kamen's website, I have to say. The structures that it's hard to find a single publication that has authoritative verified structures for all of these compounds. Myself and two of my colleagues, Mandy Moore and Donna Papson, are actually working on a monograph for the United Nations Office on Drugs and Crime right now, which is a comprehensive review of a lot of the material I talked about today, but a whole lot more. And we do have verified structures for all of these compounds in that document. So that will be available later this fall. But I can't speak right now to what's on Kamen's website, unfortunately.
All right. Next question is asking if there is a chart anywhere that you know of ranking the analogs in order of suspected potency.
So we haven't been able to find any published comprehensive chart like that. I did show one slide where we have some of the relative potency information that my colleague Donna Papson has derived from multiple different publications. We have a slightly more extensive version of that list that will appear in the UN monograph. But what is in the slide that was presented today is the most current, or the most comprehensive list that I'm aware of. And again, I just encourage people to exercise a little caution about using those numbers quantitatively. You hear all the time about the fact that carfentanil is 10,000 times more toxic than morphine. That may be true in an animal model, and it may be true with respect to analgesia, but is it really true with respect to respiratory depression? I think we don't know for a lot of those compounds. So they're a good rule of thumb, but be careful about using those relative potencies quantitatively.
All right. Next question we have is a kind of a two-part question. Do you find that Kamen is the best laboratory to use as a search tool, or are there any recommended distributors that you can recommend to purchase reference standards of the fentanyl analogs?
So we certainly have purchased and do purchase materials from Kamen, but we also publish or purchase compounds from a variety of other vendors. Cerilliant, Toronto Research Chemicals, LGC in the UK, Lipomed in Switzerland, So we monitor the catalogs of a number of different vendors. That's by no means comprehensive. I'm sure there's people I left off that list. It's rare that you'll find everything from one vendor, but their websites are usually pretty navigable, and always check more than one place. Look for the best price.
All right. Next question is from Loren Edwards. She's asking if you can comment on the ability to detect opioids after naloxone has been administered. Theoretically, the drug should be recirculating and available for detection in blood, but that's not what they're observing in their casework in Wisconsin.
We've recently worked on a collaboration with a group up in Boston looking at identifying opioids in cases where they've administered naloxone for reversals. And we're measuring both naloxone and the opioid agonists in those samples, so we haven't seen any problem with that in our experience.
All right. Next question is from Craig McKenzie. We are doing fentanyl metabolomics work in polydrug situations. For this and NMR, we are dissolving the drugs in DMSO and wear double nitrile gloves. He was just wondering if you had any comments regarding that.
Well, certainly something like DMSO is a vehicle that can enhance transdermal absorption. So it's a situation where you'd have to be careful about any skin contact. So certainly lab coats have sleeves covering your arms, any bare skin. I think double nitrile gloves is a good precaution, and if there is any visual evidence of transfer of a liquid onto the glove, to immediately remove and discard the gloves. You're not creating an opportunity there for aerosolization or dust clouds or anything like that. So I think as long as you're careful and don't have exposed skin, that's about the best you can do.
We're going to take about 5 more minutes for questions, and then please make sure to take down Dr. Logan's contact information, email there if we do not get to your question so that you can email him directly. Our next question is, in your opinion, or if you have an opinion, what are the main reasons for the production and observed prevalence of these analogs? Is it law avoidance, cheaper than heroin for illicit purveyors, or is it likely that the abundance of the analogs will decrease as laws encompass more structures and analogs?
Yeah, that's a good question. It's hard to get inside the mind of the manufacturers. Our experience with synthetic cannabinoids has certainly been that the clandestine manufacturers do pay attention to scheduling, because we frequently saw as soon as something was scheduled that a new compound would be out just within a few weeks of that. So there's probably an element of that with the opioids. I know that it's believed that a lot of this product comes from China, and the Chinese have also started scheduling. They're a signatory to the 1961 convention, so they have scheduled those compounds, but they've also started scheduling other compounds. In fact, China has scheduled fentanyl, whereas currently in the U.S. it's not specifically scheduled. We do, of course, in the U.S. have the Analog Act, which is kind of a catch-all, although it's a lot less clean than having specific compounds or classes of compounds or core components of the structure identified in the scheduling language. So I think there's an element of people trying to beat the scheduling, trying to argue that something's not specifically named so it's legal. Some of it could just be trying to find a drug that appeals, that has some kind of marketing or market advantage if in street opioid markets, or trying to find something that has a distinguishable feel or sensation or experience for the drug user. I'm sure those are all components.
All right, next question we have is, can you please provide more details on the initial validation of your Tier 2 compounds?
Sure. So when we identify something as being an emerging compound that we want to start being able to report, the first thing we have to do is, so we'll make identifications from an intelligence point of view based on analysis of the mass spec data. In addition to the TOF data, the accurate mass data, we also have QTOF resources and structural elucidation tools that we use to presumptively identify things, again, for purposes of deciding what we're going to do in terms of test development. And at that point, once we have a tentative identification, we would order the standard and verify that identification. As I said, sometimes we've gone to the length of having standards custom synthesized, but we'll look around amongst many of the vendors that I mentioned and others to try to find that standard. Once we have that and have verified the identity of it, then we characterize it on our TOF, and then it goes into that secondary database. And at the same time, we will look for a secondary confirmatory method. Traditionally, that's typically been GCMS. That, certainly for the cathinones, that's more difficult for the opioids because of issues that I mentioned related to sensitivity. So some of these we're looking now to QTOF as a secondary independent identification. So once we have it characterized in our TOF method and we have a secondary independent method, at that point we'll start we'll do multiple injections over multiple days at multiple concentrations. But for qualitative reporting, that's the extent of that initial validation. And once we've determined that something is established in the market, then we'll invest the resources of doing a complete validation according to our comprehensive method validation protocol. And typically at that point, we'll create a quantitative confirmatory test. So that's basically the process. Usually we can get that done in about 3 months, start to finish, as soon as the standard is available.
All right, we're going to take 2 more questions. The first one is from Andrea Fox asking, have you done any testing on if or what rate these substances cross-react with traditional fentanyl immunoassay tests?
We have. We've done some preliminary testing of that. Again, in the UN monograph that will be out later this year, there's some data in there. We recently did some studies on cross-reactivity in the Neogen fentanyl ELISA. We found that the 4-ANPP, which is that metabolite, but can also be a precursor. In fact, it's the typical precursor for fentanyl synthesis. Does not cross-react. But drugs like beta-hydroxythiofentanyl and norfentanyl, actually these 2 compounds did not cross-react. We did get cross-reactivity with acetylfentanyl, butyrophentanyl, furanylfentanyl, alpha-methylfentanyl, parafluorofentanyl, and 3-methylfentanyl. So it seems like with the fentanyl kits, a number of these compounds are going to give you some cross-reactivity on your ELISAs. So if you're starting to see, if these drugs are not all part of your confirmatory method, but you're seeing a lot of unconfirmed positives in ELISA, you might want to look to updating the scope of your confirmatory method.
All right, and the last question we have for the day is a 2-parter. We are seeing 4-ANPP in solid dose bulk fentanyl casework. Might this be why 4-ANPP is showing up in toxicology analysis? And what is known regarding the pharmacology of 4-ANPP?
So 4-ANPP is inactive. As I mentioned, all of the fentanyls that we looked at earlier have that phenyl group attached to the piperidine ring, and the 4-NPP has that moiety removed, so they are not they don't retain the activity of fentanyl. Undoubtedly, some of what we're seeing in toxicology casework is either unreacted starting material from clandestine synthesis. We've also had some seized drug exhibits that were pure 4-ANPP, so they were the precursor chemical that was being sold as an opioid, but in fact it was just the unreacted precursor. So there are studies that have been done with purified drug material that indicate that 4-NPP can be a minor metabolite, but I would suspect that a lot of the 4-NPP that's showing up in casework is in fact just a contaminant in the seized drug. I think that's a good observation.
All right, and with that, I know we did not get all of today's questions answered. Again, if your question was not answered, please make sure to email Dr. Logan directly, and he will get back to you as soon as he possibly can. With that, we are going to go ahead and move into our closing. Obviously, the first thing we would like to do is to thank Dr. Barry Logan for his presentation today. Dr. Logan, is there anything that you would like to leave our audience with before we close out for today?
No, I'd just like to thank everybody for participating. I appreciate all the questions that came in. If there's specifics that we didn't get to, if you email me, I will do my very best to get back to you on those.
All right, thank you again, Dr. Logan, and obviously we would like to thank all of our participants for joining us today. Without your participation, we obviously could not have these webinars as we do. So thank you very much for attending the third part of our opioid series. Make sure to register for the next parts that we have coming up. You will see again there at the bottom of your screen, we do have the PDF version of today's presentation, so make sure you download that before we close the room out. I'll leave it open for a few minutes. We also have some web links that were on the final slide of Dr. Logan's presentation. Again, Dr. Logan's email address there is in the center of your screen. We also have our customer satisfaction survey there. If you click on the word "here" in the middle of your screen, it will open up a new window for you. That will take you to our brief customer satisfaction survey where you can give us your feedback about not just the delivery of our webinars, but also the content, and we definitely value your opinion on that. You will see also in the chat pod, you might want to scroll up a little bit, we have put in our social media outlets we have there. We have a Facebook, LinkedIn, and Twitter account that are open and running now. So please make sure to follow us on all of our social media accounts. I have also opened up a new browser window for you. That is the link to our brand new website where you can find all of our at no cost webinars that we offer, as well as all of the opioid series that we are offering throughout the rest of this year. Again, thank you very much, Dr. Logan, for your presentation. Thank you to all of our audience members, and we hope everyone has a great afternoon.